picture here, we simply illustrate the fundamental organization of the kidney, and note that it's divided mainly into a cortex and medulla, with basically a hilum that faces medially. The hilum is basically the point of entrance and exit for the vasculature, renal artery and the renal vein, and the point of origin of the ureter. The renal pelvis, which is the expanded upper portion of the ureter, actually becomes subdivided as we moved into the area of the kidney into 2 or 3 major calyces that then subsequently receive several up to 8 minor calyces, and as we'll see, each of those minor calyces is associated with the apex of a medullary pyramid. And certainly branches of the renal artery vein and its nerve supply basically supplies all parts of the corresponding kidney. Pictorially, we can see the arrangement of the area of the hilum first, which, as illustrated on here, is the point of exit of the renal pelvis, the dilated upper portion of the ureter, which is basically a tube of smooth muscle. And you can see how the major calyces, 3 or 4 of them, contribute to form the renal pelvis, and how up to 8 minor calyces join together to form the series of major calyces, again contributing to the drainage of the kidney. And you can also see the apex of the medullary or renal pyramids, basically which are the site where the collecting ducts enter or exit and drain their corresponding urine into the minor calyces. The cortex, you can see, is outermost, containing obviously major components of the uriniferous tubule, the nephron, but notice we'll see that parts of a nephron are going to extend down into the renal columns between the medullary or the renal pyramids. Being more specific, we know that the cortex basically contains the major components of the uriniferous tubules, and the medulla basically consists of an array of medullary pyramids, largely containing collecting ducts, but separating those medullary pyramids will be an array of renal columns containing basically the loops of Henle of a corresponding series of uriniferous tubules. The actual nephron consists of a renal corpuscle, which is a glomerulus, a tuft of capillaries surrounded by Bowman's capsule. Then from the renal corpuscle in Bowman's capsule emanates the proximal convoluted tubule, the loop of Henle, which we can see extends down into the renal columns in the medulla. And then basically the ascending limb becomes continuous with the distal convoluted tubule. When the nephron joins with the collecting duct, that basically forms the so-called uriniferous tubule. The collecting ducts then, as we illustrate in this picture, open into one of the 8 or so minor calyces. Let's look first at the renal corpuscle, which we know consists of a tuft of arterioles provided by or containing blood from an afferent and efferent blood vessel surrounded by the elements of Bowman's capsule. This obviously is where we find the filtration barrier associated with the kidney, such that the afferent arteriole enters, providing arterial blood to the kidney, and basically it's covered by what we call the visceral layer of Bowman's capsule, which is characterized by the distinct podocytes and their foot processes. The parietal layer is continuous with the visceral layer, is outermost, and between the parietal and visceral layers of Bowman's capsule is the so-called urinary space, into which we will find the ultrafiltrated blood plasma after it's been filtered through the corresponding filtration barrier. The inset area of detail in this picture illustrates at least diagrammatically, the elements of the so-called filtration barrier. Basically, we can see a fenestrated capillary endothelium lining the afferent and efferent, the capillary bed interposed between the afferent and efferent arterioles. A shared basal lamina, and on the other side of the basal lamina we find the foot processes separated by small diaphragms of the podocytes, and then on the other side of the cytoplasm of the podocytes we find the so-called urinary space interposed between the parietal and visceral layers, the podocyte layer of Bowman's capsule. In an electron micrograph on the next slide, first at low magnification on the left, we can see an illustration of a number of podocyte nuclei and their adjacent cytoplasm. And you can see many examples of the podocyte foot processes extending down to become in direct contact with the shared basal lamina between the podocyte foot processes and the discontinuous fenestrated capillary endothelium. At higher magnification on the right side of this picture, again, podocyte cytoplasm is evident. You can see some cytoplasmic processes of that podocyte emanating down to form part of the filtration barrier. The area indicated by letter E is the lumen of the fenestrated capillary endothelium. The area indicated by B on the other side of the podocyte foot processes is the urinary space. And certainly, as we've described earlier, the major elements of a filtration barrier such as this one is the presence of a type 4 filtration collagen that characterizes the basal lamina. And as you'll hear about in pathology, much of kidney pathology is caused by changes in the basal lamina, particularly in cases of Goodpasture syndrome where antibodies are raised against the type 4 collagen in the basal lamina, or in patients that develop Alport syndrome where basically there's a splitting or thickening of the basal lamina that is similar in a disease process to one that also affects the stria vascularis, which is the site of where endolymph is produced in the inner ear. Now let's kind of summarize functionally what the different components of the nephron distal to Bowman's capsule are involved in, beginning with the proximal convoluted tubule. As indicated here, it's certainly the longest, most convoluted segment of the nephron, and basically it's where much of the glomerular filtrate is resorbed back into the proximal tubule. And basically, this has a dramatic surface area characterized by a tremendous brush border and a large surface area specialized for resorption and secretion, respectively. As we move down into the loop of Henle, the loop of Henle forms a hairpin loop of the nephron that extends down into the medulla and consists of thin and thick segments. But the descending and ascending limb have distinct differences relative to their permeability to water. The descending limb is permeable to water and less so to urea and sodium chloride, such that the fluid at the bend of the loop of the Henle, deep in the medulla of the renal column, is hyperosmotic relative to blood plasma. The ascending limb is impermeable to water, but permeable to sodium chloride and urea. So therefore, by the time the fluid reaches the distal tubule, it is now hypoosmotic relative to blood plasma. In the distal convoluted tubule, it's lined by a more cuboidal epithelium whose major job is to resorb sodium chloride, but now not urea, from the tubular filtrate. By the time we reach the collecting tubules, basically these are the part of the nephron that are basically under control or respond to antidiuretic hormone or vasopressin, that's the posterior aspect of the pituitary, and vasopressin regulates the permeability of the collecting tubules to water. So therefore, the collecting tubules become kind of the final common denominator in regulating water conservation and the concentration of the resultant urine. Finally, let's look at some specialized cell types that are associated, again, back at the level of the renal corpuscle. These are mesangial cells that are either intra or extraglomerular. What's illustrated here are some extraglomerular mesangial cells, and some specialized cell types that are known as juxtaglomerular cells that are basically lining the lumen of the afferent arteriole, and the adjacent macula densa cells, which are taller, more columnar cells in a closely associated wall of the distal tubule. Functionally, the juxtaglomerular cells are basically modified myoepithelial cells, which we've seen in the wall of the afferent arteriole. Their job is to secrete renin, which basically converts circulating polypeptide angiotensinogen into angiotensin 1. And then subsequently, AT1 is converted to AT2, which is a potent vasoconstrictor that stimulates aldosterone secretion from the adrenal cortex. And certainly, as we know from physiology, aldosterone increases sodium and water resorption in the distal portion of the nephron. The macula densa cells, as we indicated, are taller, more columnar cells in the adjacent distal tubule, and basically their function is thought to sense the sodium concentration in the area of tubular fluid. So now this is a change in the notes. We identified these cells in the notes as being pulcescent cells, but now we know much more about them than was originally stated in the notes. These extra and intraglomerular mesangial cells are actually parasites, which in our case, as we illustrated previously, are found between the afferent and the efferent arterioles near the vascular pole of the glomerulus. These cells we now know are acting as parasites, are contractile, phagocytic, they are capable of synthesizing collagen, and they produce factors that induce constriction of the glomerular arterioles and therefore can regulate the filtration rate by changing the luminal diameter of those arterioles. So they have much more of a functional significance than we previously thought. Finally, a comment, and I've added this as well, about erythropoietin. Erythropoietin is the major regulator of erythropoiesis, logically, and it is a glycoprotein that's produced mainly in the kidney. And what can come up on the exam is what kind of cells and where is this produced. As we know now, it's produced by what are known as peritubular interstitial cells that are largely found in the inner part of the cortex of the kidney and in the outer part of the medulla.